Introduction. Welcome, students. Today, we will embark on a detailed journey into the world of retinoid metabolism and its impact on cellular processes. Retinoids, which are derived from vitamin A, play crucial roles in various biological functions, including vision, development, and cell growth. In this script, we will explore 12 key topics related to retinoid biochemistry, from absorption to signaling pathways. So let's dive in. Absorption is the first step in retinoid metabolism. Retinol and retinol esters, the predominant dietary forms of vitamin A, are absorbed in the small intestine. The process involves multiple steps, starting with retinol ester hydrolysis by pancreatic lipases. Once hydrolyzed, free retinol is solubilized by bile acids and forms micelles to facilitate its uptake by enterocytes. Within enterocytes, retinol is then esterified by lecithin retinol acyltransferase to form retinol esters for packaging into chylomicrons. These chylomicrons enter the lymphatic system and eventually reach the bloodstream, where they distribute retinoids to various target tissues. To ensure efficient transport of retinol in the bloodstream, it must be bound to retinol binding protein, RBP, in a tightly regulated manner. RBP is synthesized and secreted by the liver, where it forms a stable complex with retinol. The RBP retinol complex interacts with transthyretin, TTR, which prevents renal filtration and enhances retinol stability. TTR also aids in delivering retinol to target tissues, where it can be released and taken up by specific cell surface receptors. At the cellular level, retinol uptake is mediated by cell surface receptors, such as stimulated by retinoic acid 6, STRA6. STRA6 facilitates retinol binding and internalization into cells. Intracellular trafficking of retinol involves its association with cellular retinol binding protein, CRBP, which protects the molecule from oxidation and guides it to specific retinol metabolizing enzymes within the cytoplasm. In addition to immediate utilization or conversion, retinol can be stored as retinol esters in hepatic stellate cells, HSCs, within the liver. HSCs possess lipid droplets, which serve as depots for retinol ester storage. This reservoir of retinol esters can be mobilized in response to physiological demands, such as cellular differentiation or tissue repair. One of the key steps in retinoid metabolism is the conversion of retinol to retinol, which is catalyzed by retinol dehydrogenases, RDHs. RDHs are a family of enzymes that oxidize retinol, resulting in the production of retinaldehyde. This reaction is reversible and plays a vital role in maintaining the balance between retinol and retinol pools in cells. Retinaldehyde exists in multiple isomeric forms, with the all-trans isomer being the most common. However, in order to fulfill various biological functions, retinoids often require specific isomeric configurations. Retinoid isomerases, such as RPE65, mediate the isomerization of retinol, allowing its conversion from all-trans to 11-cis isomer. This conversion is crucial for vertebrate vision, as 11-cis retinol forms the chromophore of rhodopsin. The synthesis of retinoic acid, RA, involves the oxidation of retinol by retinaldehyde dehydrogenases, RALDs. This irreversible reaction results in the formation of RA, which serves as a biologically active form of vitamin A. The biosynthesis of RA occurs in various tissues, including the liver, kidneys, and developing embryos, where it plays essential roles in cellular differentiation and embryonic development. To facilitate the intracellular transport of RA, it binds to cellular retinoic acid binding proteins, CRABs. CRABs enhance the solubility and stability of RA, preventing its degradation and facilitating its delivery to target nuclear receptors. The binding of RA to CRABs is reversible, enabling regulated release and uptake by specific cellular compartments. Within the nucleus, RA binds to specific nuclear receptors, known as retinoic acid receptors, RARs, and retinoid X receptors, RXRs. These receptors function as ligand-activated transcription factors, regulating the expression of numerous target genes involved in cellular differentiation, proliferation, and homeostasis. The binding of RA to RAR-RXR heterodimers initiates a cascade of transcriptional events, resulting in cellular responses to retinoid signaling. The effects of RA signaling must be tightly regulated to maintain cellular homeostasis. RA undergoes metabolism and clearance through various pathways, such as oxidation by cytochrome P450 enzymes and conjugation with glucuronic acid for excretion. These metabolic processes prevent excessive RA accumulation and ensure its controlled availability for cellular processes. The binding of RA to RAR-RXR heterodimers initiates a cascade of intracellular events, leading to transcriptional regulation. This includes the recruitment of co-regulatory proteins, chromatin remodeling, and modulation of gene expression. This complex interplay allows for precise control of cellular responses to retinoid signaling, ensuring proper development and differentiation. Retinoid signaling pathways are involved in diverse cellular processes, particularly cellular differentiation. RA signaling plays critical roles in the development of various tissues, including the eye, limb, and central nervous system. It regulates the expression of key genes governing cellular fate determination and morphogenesis. Disruptions in retinoid signaling can lead to developmental abnormalities, highlighting the importance of this pathway in embryogenesis and tissue regeneration. In conclusion, retinoid metabolism is a complex process involving absorption, binding proteins, intracellular transport, enzymatic conversions, and transcriptional regulation. The diverse roles of retinoids in vision, development, and cellular differentiation highlight their significance in human biology. Understanding the intricacies of retinoid biochemistry provides us with valuable insights into potential therapeutic approaches for retinoid-related diseases and disorders. As future medical professionals, this knowledge will be essential in providing optimal patient care.